these damaged DNA, these cells containing damaged DNA in the circulating blood, significantly reduces it, like here. So does exercise. And so does a combination of both of them. So some mice actually exercised and got CT scans. It all shows that the spontaneous level of your spontaneous genomic instability is suppressed by exercise or by CT scans. So again, suggesting that both exercise or CT has a beneficial effect to the whole organism. If we then take the organism and challenge it, or the mice challenge it with a large dose of radiation, in this case we used 1 gray gamma radiation, and then compare the effect of this exercise regime or the multiple CT scans or a combination of both, we have suppression of the 1 gray induced micronucleated reticulocytes in all the groups, but we only have significance in the group that exercised or got exercise in CT scans. Although you can see a suppression in this one here. It did go down, but not significant. So again, it's saying to us that exercise or exposure to low doses of diagnostic radiation has some kind of adaptive response that confers a protective effect on mice. If we look at gamma H2AX levels in the bone marrow cells of these mice after their exercise or CT scans, we can see a similar effect. I don't see anything at 0 gray, but this endpoint is looking for sites within the DNA that have been damaged. And you can see that after we give a large dose of 1 gray or 2 gray, that the mice that had nothing done to them had these levels in green bars, but the ones that were exercised or got CT scans had a suppression, again suggesting that some kind of protective effect. The interesting part about this endpoint is when you combine both modalities, that protective effect goes away. So for this particular endpoint, a slightly different result. We are seeing suppression with the two exercise or CT scans. The third endpoint, apoptosis, again gives the same result. If you look at what happens when you give a 1 gray or 2 gray dose to these mice, which are the green bars, you have a higher level. When you compare the same dose in mice that have either had exercise or CT scans or both, at 2 gray we get significance in all 3 groups. At 1 gray we get significance in the combined group. And at 0 gray we have suppression in all 3 groups again. So again, we're seeing something that's very common between exercise and diagnostic radiation that's inducing adaptive responses that suggest to us that perhaps we are having a protective effect of both modalities. We also did an acute study, which basically is looking at a single large dose of radiation or a single bout of extensive exercise in mice. So 1 CT scan compared to get a couch potato mouse and run the crap out of them for 1 hour. See what happens. And if you look at the study we looked at, if you compare apoptosis, in this case, this single bout of exercise produced a massive amount of free radicals in an unconditioned mouse that caused a lot of apoptosis to happen, even in the absence of radiation. And if you look at increasing doses of radiation, both CT scans and exercise, given acutely once, enhanced the level of apoptosis. Well, if you think about apoptosis being an anti-carcinogenic process, even single bouts of exercise in this particular situation may be beneficial, but we're not sure. So I have to conclude now at the end of this. What time is it? It's about on time. Any questions before I go on from the audience? Nothing?
Sure. I think I have one. Well, I haven't shown a lot of stats about. Do we have like demand?
Yeah. So the question is, we're showing data that suggests that we're having protective effects of low doses of radiation or even exercise in this particular experiment. And is there other labs out there that are looking at mechanisms to this? In fact, the answer to that is yes. There are many labs looking at mechanisms. We had a mechanism of the role of p53 in this. We're looking at how p53 can influence, can modulate this process of adaptation. If you look at cellular studies, if you look at in vitro and in vivo studies in animals, you can see that there are a number of different mechanisms. One is that enhancing apoptosis reduces cancer risk. So a lot of labs are looking at how apoptosis can be modulated or modified by low doses. So enhancing the number of cells that die represents killing more cells that could potentially become cancerous. So it's an anti-carcinogenic process. Other labs look at DNA repair, and you can take repair mutants that have different pathways that are inhibited and say, if I do this low-dose radiation and turn on a protective mechanism, if I take away a DNA repair pathway, does that affect it? it looks like in, say, lower eukaryotes like yeast, it's all to do with homologous recombinational repair. So repairing DNA double-strand breaks. And if you make a defect in that pathway, well, the cells won't adapt. So there are many labs looking at mechanisms of this. There's cell-to-cell communication. Lots of labs are looking at how some cells talk to others. So if this cell gets irradiated, sends out a signal, it can cause other cells to commit suicide, to protect the whole organism. So yeah, there's lots of studies on that.
Are all the tests done, and so the mice are exposed?
Yeah, so no, the answer to that question is no. The question is, are all the challenge doses done, all the challenge doses done in vivo or in vitro? Yes, so the 1 grade, the 2 grade, so you can do it a number of ways. You can look at taking the cells out of the organism, giving them a low dose exposures, the priming doses, and challenging them in vitro. Or you can do the whole mouse, prime them, and challenge them in vivo. And you can see similar results for all this stuff. We typically will do endpoints that require the mouse to actually go through some metabolic process like the micronuclear reticulocyte assay, where we irradiate the mice, do our priming doses and challenge doses, but we have to wait 2 days for the damage to manifest itself in the circulating blood. So that obviously has to be done in an in vivo situation. But other assays like apoptosis, we take them out of the mouse because if the cells are committing suicide in the mouse, they will be absorbed or resorbed by the mouse, so we won't see them in circulating blood. Which is why when you do look at apoptosis in humans, you typically would take the blood out of the human after you did a whole body, say, scan, a diagnostic scan. You take the blood out, and that way you can keep track of the cells that are actually dying in vitro. Because otherwise it would just disappear in the blood. You would have no way of telling which ones are dying through apoptosis, which aren't. So it depends on the endpoint what you're looking for. Other question?
So if the adaptive response is more like a slow photosynthesis-dependent process, then if you were to give them a lunch dose of 1 normal liter, then we still see that same response, given that these proteins might already be.
Yeah, so that's a good question. So if you look at the adaptive response, the question is, we look at particular times after a priming dose to turn on the adaptive response to see the biological effect. And we call those our short-term studies, right, to look at where we see an effect. And if we see an effect, we can say, okay, something happens maybe at 6 hours with this pathway, and maybe at 12 hours with this pathway, and maybe at 24 hours with this pathway. We know we're changing something with the low dose. Some experiments look at long-term effects. Like our lifetime study, we'll be looking at, if you do something at 6 weeks of age, how does that change your risk of getting cancer a long time down the road? We don't know if it's changing the risk at the time of the radiation or whether or not weeks after we experiment, something is going on. So we've got a group of mice in this particular experiment that's actually looking at the effects of how we can turn on this adaptive response later on for longer periods. There's research out there that shows that up to 3 weeks, some mice will actually have inducible radiation resistance conferred on them. So if you do a survival curve in mice and look at the lethal effect of, say, 7 gray of radiation, most of those mice after 7 gray will die in 30 days. So LD 130. If you give mice a whole body priming dose, right, and then give them the 7 gray, you can wait up to 3 weeks. And these mice have 80% survival. So you can show that this response can actually confer resistance up to 3 weeks or even longer. I've seen some studies that say 6 weeks. And so it depends on your endpoint and what you're looking at, but that's why these experiments are are very costly and time consuming, because you're looking at one challenge dose, one priming dose, and you can look at the times in between. It can go on forever. So, question? So
this adaptive response suggests then that having low-level free radicals produced actually makes the organism more resistant to any sort of future stress, right?
Yep.
So it's common that blood are to consume free radical or to consume antioxidants.
Yeah.
In doing so, could you essentially be yourself more sensitive to potential?
Yeah, there's a big debate on that right now. Yeah. In fact, eating your antioxidants. The question was, there's lots of people that believe eating your antioxidants in your diet will actually protect you from effects of free radicals. But if free radicals induce a protective mechanism, by reducing the levels, is that going to have a negative impact, right? And I think there is a debate on that. I don't know. I know that. Let's look at it another way. If you take away endogenous free radicals, is that good for an organism? The answer to that question is no. You know, if we look at studies where you take away natural background radiation on living organisms, they don't do so well. So it suggests that you need a certain level of reactive oxygen species or free radical damage to maintain your systems and keep them sharp. I suspect if you overdid it on the antioxidants and lowered your free radical levels, it would be very similar to going to low background areas, and that might give you the same effect. On the other hand, if you're going out and running a marathon and producing buckets of these things, so again, not in moderation but in excess, then taking your antioxidants would be a very good thing to do. If you look at a marathon runner, after they've done a marathon, you take their blood samples out and look at DNA damage in their cells, like do a comet assay on them, their DNA is all beat up, all beat to hell. So it would probably be a good idea to have an antioxidant shake after a marathon, or maybe before. Right? So I guess we'll just talk, I'll just do a few conclusions here. It's, from this particular research I talked about today, we can say that routine exercise and repeated CT scans, even though the dose gets high after repeating it, does not cause an increase in the spontaneous damage to cells, at least by the endpoints we have. It doesn't increase apoptosis, it doesn't cause chromosomal damage, and it can induce an adaptive response, which suggests to us it could have an anti-cancer or reducing cancer risk. There are differences that exist between chronic and acute treatments, and there are big differences between the endpoint we're looking at, whether we suppress the process or we enhance the process, depending upon which one we're looking at. And this is just a picture of our animal facility, which is, I understand, was complete chaos, but now since we're halfway or over halfway through the study, it's not too bad anymore. And obviously this work couldn't have been done without a lot of hard work from a lot of people, grad students and technicians included. And we managed so far to pull it off, and I suspect that we will pull this off and glean some really good data about cancer risk in cancer-prone animals and the effects of diagnostic radiation on it. These are just acknowledgements. Again, I said this is paid for by the U.S. Department of Energy. People in my group, Nifan, Christina Taylor, Mary Ellen Zabowski, and Nicole McFarland were the technicians, and Lisa Laframboise. We got a lot of help from the McMaster Small Animals Imaging Facility, as you can imagine. Multiple hundreds of CT scans and PET scans that were done. And that's about all. Any other questions before we better down? See ya. Yeah. Good group.